Have you ever wondered how cells ensure that all chromosomes are correctly attached before they divide? Well, that's where the metaphase checkpoint, also known as the M phase or spindle checkpoint, comes into play. This checkpoint is a crucial stage during mitosis, the process of cell division. Its main job? To check if all the sister chromatids, those identical copies of DNA, are correctly attached to the spindle fibers. You see, the checkpoint ensures that all the chromosomes of cells moving into the next stage, called anaphase, are firmly attached to at least 2 spindle fibers from opposite poles of the cell. This is absolutely vital because the separation of chromosomes in anaphase is an irreversible process. Any mishap here, and it could lead to serious errors in cell division. So it's clear that the metaphase checkpoint plays a crucial role in cell division. But how does it work exactly? The metaphase checkpoint is not just a simple gatekeeper, it's a complex mechanism that involves several proteins. Now let's dive into the heart of the matter. The metaphase checkpoint is like a vigilant guard, ensuring that all sister chromatids, or identical halves of a duplicated chromosome, are correctly attached to spindle fibers before the cell moves into anaphase. This is a critical step because once the chromosomes separate in anaphase, there's no turning back. So, how does this checkpoint work? The key lies in a group of proteins that keep a sharp lookout for any straggler chromosomes in the cytoplasm. These proteins are the unsung heroes of cell division, working tirelessly to ensure that every single chromosome aligns perfectly. If a straggler chromosome is detected, these proteins spring into action, halting the cell cycle until the issue is resolved. One of the major players in this process is CDC20. This protein is negatively regulated by the metaphase checkpoint. In simpler terms, the checkpoint puts a brake on CDC20, preventing it from activating the ubiquitin tag. Now you might be wondering, what's this ubiquitin tag? Well, it's a molecular signal that triggers the anaphase-promoting complex, a key player in moving the cell from metaphase to anaphase. By preventing the activation of the ubiquitin tag, the checkpoint ensures that the cell doesn't rush into anaphase before it's ready. But the checkpoint's job isn't done just yet. Once all chromosomes are correctly attached, the checkpoint needs to be deactivated. But how does this deactivation occur? One of the main mechanisms is by transporting motor complex proteins away from the kinetochores, which are the points of attachment between the spindle fibers and chromosomes. These proteins are then redistributed to the spindle poles, effectively turning off the checkpoint and allowing the cell to progress into anaphase. So, there you have it. The metaphase checkpoint is a fascinating example of the intricate coordination and regulation that underlies each and every cell division. It's like an incredibly complex ballet, with each dancer knowing exactly when to move and when to pause, ensuring the performance goes off without a hitch. The metaphase checkpoint doesn't stay active indefinitely. It's deactivated once its job is done. The metaphase checkpoint, often called the spindle checkpoint, is a critical point in the cell cycle. It ensures that all chromosomes are correctly attached to the spindle fibers before the cell moves into anaphase. But what happens when all the chromosomes are correctly attached? How does the cell move forward? Here's where the deactivation of the metaphase checkpoint comes into play. The metaphase checkpoint is deactivated to allow the cell cycle to progress. And this deactivation occurs once all the chromosomes are correctly attached. So, how is this deactivation achieved? It's all about the proteins, the motor complex proteins, to be precise. These proteins play a crucial role in this process. Once all the chromosomes are securely attached, these motor complex proteins are transported away from the kinetochores. The kinetochores, remember, are the points where the spindle fibers attach to the chromosomes. Now, where do these proteins go? They are redistributed to the spindle poles. This redistribution is a sign that all is well, that all the chromosomes are aligned and ready for the next stage of the cell cycle. This deactivation of the metaphase checkpoint is a vital step in cell division. It's like a green light, signaling that the cell is ready to move forward, ready to divide. Understanding the metaphase checkpoint is key to understanding the complexities of cell division. It's a fascinating process, isn't it? The metaphase checkpoint is more than just a simple check and balance system. It's an intricate process that ensures each sister chromatid is correctly attached to the spindle fibers. This checkpoint prevents premature anaphase, a stage that is irreversible, by negatively regulating CDC20 and halting the activation of the anaphase-promoting complex. Once all chromosomes are correctly attached, the checkpoint is deactivated through various mechanisms, including the transport of motor complex proteins away from the kinetochores. The metaphase checkpoint ensures the accuracy of cell division, making it a crucial part of the cell cycle.